different gene after you're done.
That sounds good. So I'm going to start up again from the top just to make sure we're all on the same page. So I picked the first gene in group number 4. And again, I just oriented everybody. We're looking at Toxoplasma gondii ME49. It's chromosome 7A. We are in the sort of the right arm of the chromosome. So if you zoom into this, if you look in this region, you'll find that these genes are present in this region. There's some red ones and blue ones. Hopefully everybody's now familiar. The red ones are right to left and the blue ones are left to right. Obviously, relative to the orientation of a chromosome, you could flip the chromosome and then the reds and the blues will be flipped as well. So we talked about this. Oh, one thing I highlighted is that there's a really cool feature in the JBrowse where you can pin a track to the top. And that's what I've done with these 2 tracks right here, which means that they stay at the top of the page as you scroll up and down. So that's really nice if you're looking at multiple tracks and you want to see the gene model in front and center as you do this. The first, and let me actually make sure this is being, yeah, it is going. So the first thing we looked at is this transcription start sites. So this is a data set that specifically looks at the sequence of the 5' transcript start sites, right? Not translation start site, the transcript start site. So these are typically will include the beginning of where transcription starts, where the UTR starts, the untranslated 5' region. And each one of these, if you click on these, it'll give you a score. I actually I couldn't remember if this score is a good score or a bad score. And so I went to our About This Track option. And as you can see here, each feature has a score of 1 to 6, 1 indicating the highest score. So 2 is a pretty good score, I guess. And so it gives you a little bit of confidence about these transcription start sites. And so if you are interested in making sure you have the exact transcription start of this gene, this track may be useful to you. Of course, like with any data, this is data that was generated in a lab. You have to consider the strain that was used in the experiment, consider the experimental conditions, the developmental stage, depending on your organism, of when the sample, the data was collected. So all of these things you have to factor in as you explore these tracks. Okay, moving down, we have this RNA sequence track data. And this data is called RNA-seq evidence for introns. So each one of these bars highlights an intronic region. I'm just going to jump here to where I'm zoomed in. And you can see that there is, each one of these bars corresponds to a possible intron. And in many cases, these introns match the annotation, right? You can see there's an intron here and there's a bar in the intronic region. There's an intron here, there's a bar in the intronic region. So that's pretty good. So how is this data generated? And Suzanne already said this, I'll just repeat it. As you know, with RNA sequence data, you're sequencing the messenger RNA, the already, hopefully, mostly spliced RNA transcripts. And then the RNA, which is all these, in most cases, you're doing Illumina reads, so they're short reads. You take all of these reads and you map them back to the genome. You're not mapping it back to transcripts. You don't have the sequence of all the transcripts sitting around in a file. Typically, you have a genome. And so you map them back to the genome. And so let me double check if I have an image that can show you this. And so if you take a look here. So yeah, actually, I thought I had a better illustration, but I don't, so I won't show that. So basically, the reads that map to an exon will map nicely, and you'll get these peaks. And so if I scroll down to the bottom where I've loaded some RNA sequence data, you see this is these are called coverage plots. So this is a histogram illustrating where all the reads align to the genome. And so if I go ahead and pin this track to the top, and so now when I scroll down, you can see that this is a pretty nice match. There are reads here at the 5' end of this genome. Then there are reads here that match this exon, and these reads map this exon. And so that's great, right? But remember, in the RNA, these exons will be spliced with each other. And so you will have a number of reads that come from the junction where these two exons were spliced with each other. Now, when you try and map these reads back to the genome, these reads have to be split, right? Because the exons are split, they're not spliced together in the genomic sequence. And so when you split them, anytime a read has to be split, that's evidence that there's a possible intron in that region, okay? And the more evidence you accumulate for that region, the higher confidence you have that there is a real intron in here. And so if I scroll back up to this one here, so this bar is a representation of all the reads that had to be split between these two exons. And now if I simply mouse over it without clicking, you'll see it says reads 4,145. So it's telling me that there are 4,145 reads that support this intron. You can get more information about each of these reads by clicking on it. And when you click on this, it's going to load a table. And this table shows you some statistics up here, which may interest you. In particular, percent of most abundant intron. So it represents 66% of the most abundant. So we take the intron that has the highest number of reads that support it, and that's 100%, right? So 4,100 is 66% of whatever was 100% of that. So probably something like 7,000 reads. So that gives you an indication as well. That's a nice confidence scale, in particular when you start comparing possible alternative introns. And then scrolling down, you'll see here there's a list of all the samples from all the experiments that were used to generate this track. Different developmental stages, different experimental conditions. And so you can actually take a look at this and see where most of these reads are coming from. And sometimes you might find out that a particular intron has a lot more evidence from a particular developmental stage and may indicate if there is alternative splicing happening, that may indicate that there is a stage-specific splicing, and things to consider as you take a look at this data. So, and as I indicated before, in the intron, there are two subtracks. One is called matches annotation, and one is called unannotated with strong evidence. And as you see here, the top track, these are the ones that match the annotation. And as I scroll down, you'll see that they're all thousands of reads support these introns, and they're all fairly equal. This one, I think, is the highest one, 5,422 reads that support this intron. But you'll notice here, there are a couple of other introns that had strong evidence, but they are not part of the official annotation. And when you mouse over these, you will see that there's one of them has about 500 reads, and the other one has about 700 reads. And then this one has 3,325 reads. Okay, so now the question becomes: is there possibly another exon in this intron right here? Is there an alternative splice variant of this gene where this happens? The answer is probably yes. There probably is an alternative splice variant of this gene, but based on the numbers, based on the developmental stages and samples that were assayed, it looks like this is a rarer species than all the other ones. But it's not zero, right? There's still hundreds of reads that support this possible alternative splice variant of this gene. So again, if you were working on this gene, I would say, I would, put a little note in my lab book indicating that I should keep this in mind, because maybe there is some functionality that's encoded in this little exon here that could be potentially very, very critical or important for this particular gene function. Okay. David, you put a chat in the thing. Do you want to move to another gene? That's fine. I'm happy to let you do that.
I just think that I just think that for some other genes, we'll see some of the same things, but there's some others with some more interesting features that we might want to move to.
Perfect. You want to do that?
Yeah, I'm happy to do that. And in light of the in light of the time, I also want to suggest. So I will look at one gene. It's just the gene that my group was looking at a moment ago. But I will also encourage Uli that, given the shortage of time, if you would like to pick a particular gene or two to be able to work on re-annotation in Apollo when we hand the microphone back to you. You can just be prepared for that. So
David, a couple of things I want to make sure that in your description to explain what ChipChip and ChIP-seq data are and how they
Yep.
can help. In addition, if you could turn on
Yep.
the nanopore data and explain what that is and how that's useful. Perfect.
Have already, both on my list of things to do. So let me just do these are going to be and that's not what I meant to do. All right, so we are looking at a different gene now. A gene that my group had been looking at, one of the ones in our assigned list. And I should mention, I know there was a lot of discussion about things not loading properly. We highlighted that this might be a problem because 200 people are trying to do the same thing at the same time. In our group, we just reloaded the page a couple of times and had no problem being able to visualize everything that we were wanting to look at. But that may not always be the case for everyone. Let me just change this to a linear scale and change the score range to a local normalization. Okay. Oh, sorry, one more that I forgot to change. Linear scale, change the score range to a local. All right. Here's another gene. And the first thing to mention is this display that you're looking at looks a little different than the display that Omar was showing you. And if I can find my chat window. This is an alternative configuration that I was working on. Some of the things we talked through in iGroup. I've added some other tracks. I've configured tracks differently. I've dragged some of them around in various different places. I've added some other features down below that I'll highlight. It looks like the orthology track didn't load. I could reload to pull that up, but that's not going to be particularly important for now. I've pasted in the chat a link to the view that I have used, which you might find useful for this for this genome. And I should also mention that, of course, while these exercises work, these exercises are focused on toxoplasma genes. The same kind of queries, the same kind of principles apply if you're interested in assessing the gene models in Fusarium or Anopheles or whatever other species you might be interested in. So for this gene, I'm taking a look at the organization of this gene. Omar highlighted the value of these introns, and I should also say that the fundamental part of our message, our take-home message for today, is biology is complicated, something that we all know, right? So we would all like to have the answer of what this gene model ought to look like. And this view here is a pretty good approximation of the answer, but it is still only an approximation. There are, for every gene in the genome, there are alternative transcripts that start upstream or downstream and end upstream or downstream and are alternatively spliced and are modified in various different ways. The question of whether that should be annotated as part of the official annotation is a judgment call, and we'll see some examples of that in a moment. But ultimately, if this is a gene that you are particularly interested in, you will want to be sure that you are aware of those issues. So let's take a look at this gene. We can see that this gene is annotated to start about here and stop about here. and we can see that the start site is consistent with the CAP selected sequencing experiments arguing for the time of the start. And the same is true for long read RNA-seq data from nanopore sequencing. I have collapsed this track here just to save vertical space. But if I decide to show that better where it is here, if I decide to expand that a little further, I can see that this start site is consistent with many long reads, but there is actually a range. Few genes are actually transcribed starting from precisely one nucleotide, and similarly, these transcripts all start in approximately this region, but not always at precisely the same nucleotide. And whether that is a case of degradation of the messenger RNA, which has led to erroneous determination of the terminus, or whether this is a true polyadenylation site, we can't know from this experiment. But my guess is that, in fact, this is genuine biological data. And if I collapse the long read mRNA-seq data, again, we can start to see that this is, whoops, that was not collapsed. We can see that this is consistent with individual experiments. Now, I have also, in addition to the same tracks that were turned on in the demonstration, I've also turned on one additional track that I particularly like using. This is a track which, under the transcriptomics site, is not strand-specific RNA-seq data, but it shows transcription from 29 different strains that span the diversity of genetic variability in toxoplasma worldwide. And rather than displaying 62 tracks, which would be a lot to scroll through, I'm using these multi-density plots here, looking only at uniquely mapping reads. And so I can very quickly and easily see that the pattern I'm observing here is observed for all of these strains, right? All of these strains show transcripts start about here, stop about here, and 1, 2, 3, 4, 5, 6, 7, 8, 9 introns consistent with the annotation. So to a first approximation, this annotation looks pretty good. But as we look deeper, we see some additional information. One of the questions that we were discussing when we left the breakout room was that these first two introns show pretty substantial transcription through the intron. And intron read-throughs are common and a well-recognized feature in eukaryotic genomes, not universal. This intron here, for example, shows no evidence of intron read-through. This very little evidence of intron read-through, but these two introns show substantial evidence of intron read-through. I'll point out that these particular graphs are represented on a log scale. If I were to change this to look at a linear scale, I see a very different picture, right? On a linear scale, and for full disclosure, in order to do that, I've had to change the scoring range to 0 to 100. I'd done this previously. What did I just do? Not quite sure. Well, let's take a look at this one here. If we change this to look at a linear scale, oh, I must have changed the score range to go from, okay, there we have it back, and I'm not quite sure why that happened here. Okay. So if I change this to look at a linear scale, I can see the difference. This is just a visual representation. I'll change it back to log scale. Even here, I can see this because a mouse over shows me the depth of coverage, and I can see that while this first exon was sequenced to a depth of about 100 reads, this intron was sequenced to a depth of only about 20 reads. So 80% of the introns were excised here, or this intron was excised 80% of the time, but not 100%, as opposed to here where it was excised greater than 99% of the time. So what does that mean? That means there's a fairly high frequency of read-through of this intron. And if I take a look at that intron, by clicking on it, I can see the length of the intron, which I'm going to need to add up manually. That intron will be 514 nucleotides long. If we add one, because we need to include the first nucleotide, that's 515 nucleotides. That's not divisible by 3. So sequencing through this intron would lead to a frameshift mutation, and there may also be stop codons within that. So this would truncate the expression of this protein, assuming the initiation site is annotated correctly. So my guess is that roughly 15% of the time this intron is not excised, that probably has no functional consequences and probably should not be annotated as a non-functional RNA, but that's a judgment call. And if you are working on this gene, you should be aware of that in case there is additional evidence that suggests this may be important. Same thing applies for the second intron. We can also see that there is evidence for introns that span this exon. So this is an exon skip variant that removes this exon. It's a tiny exon. If I click on that, I can see that this second exon here is only 7 plus 1, 8 nucleotides long. I've added 1 because we have to include 83. So 83, 84, 85, 86, 87, 88, 89, 90 would be 8 nucleotides long. Removing 8 nucleotides would cause a frame shift in the protein coding sequence. So again, despite the fact that there is evidence of exon skip polymorphisms, the abundance of that is very low. Roughly 1,000 reads that support this exon skip variant versus 8,000 reads that support the exon inclusion variant. And combined with the fact that this would lead to an untranslatable protein, mean that I would be inclined not to annotate this gene, this alternative, as an alternatively spliced variant. But let's move downstream, and we can see that if we look at introns 3 and 4 spanning exon 4, see that while intron 3 is excised 8,600 times and intron 4 9,500 times, there are roughly an equal number of equal amount of experimental support for skipping this fourth exon. So here, the frequency with which this exon is skipped is roughly the same as the frequency with which this exon is included. So I would argue that we should add an additional transcript that removes exon 4. Let's look at exon 4. Here is exon 4. Exon 4 is 120 plus 1, 121 nucleotides long. That would also introduce a frameshift mutation. So this exon would likely lead to a dysfunctional protein. But the fact that half of all transcripts lose this exon suggests to me that that should probably be annotated here. Again, this is a judgment call. It's likely to be non-functional, but of course there are many reasons why a non-functional protein, a non-functional enzyme, protein phosphatase here, could still be a functional transcript that leads to, for example, a dominant negative regulator in a multi-subunit protein. So all of these issues and every issue that we can discuss today is subject to lots of gray zones for consideration, and is what makes annotation so hard. But it is also why it is imperative that, in addition to doing what all of us do, which is to look at the existing annotation and say, oh, I can see that this N-acetyl glucosamyl transferase is a gene with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 annotated exons. That's the truth. And there is no strong evidence contradicting that. For other genes, there is significant support for alternative views, and we all owe it to ourselves to make sure that we don't miss out on important biological information. Now, having described that, let's take a look at some of the other information, including information that Omar had mentioned. Let me take a look at the long-read RNA-seq data. And what I can see from long-read RNA-seq data is that despite the fact that these are supposed to be full-length transcripts, most of them probably are not. Even in this nanopore experiment, the polymerase often fell off before it got to the reverse transcriptase often fell off before it got to the 5' end. I can see evidence of frequent intron read-throughs. Let's expand that a little further to make it easier to see. I can see evidence of frequent intron read-throughs, as we'd seen in the bulk RNA-seq data, not in all introns. I can see evidence for exon skip variants, particularly in this exon 4, which is sometimes present and sometimes not. So the long-read RNA-seq data supports what we're looking at, but is not a magic bullet solving all of our problems. There were also questions about epigenetic marks, and we're including a variety of epigenetic marks here from chromatin immunoprecipitation and sequencing, or chromatin immunoprecipitation and array probing experiments. In this case, these are antibodies to, for example, a trimethyl activation mark on histone 3 lysine 4, which is associated with activation. DNA was isolated, and without removing histones, was fragmented and then precipitated with an antibody to this histone modification. Then the DNA extracted and sequenced, and either hybridized to an array or sequenced here in bulk data. And what we can see is a clear peak consistent with the transcription initiation site. But this is typically true for epigenetic marks, that the level of resolution is nowhere near the level of resolution that we would like to define a precise initiation site. We can say for sure that the data is consistent with transcript initiating here. We can say from these H3K4 monomethyl marks that this appears to be an open region that is likely to be protein coding sequence. But this is not going to help us refine our annotation to the nucleotide to the nucleotide level. So let me leave it there for this particular gene and ask if there are any questions that have come up. I see some comments in the chat from Richard, who we were talking to previously, asking about a track about intron-exon junction prediction, including alternative sites. So yes, let me highlight. So these intron spanning reads are probably the most informative information. This is not a prediction. These are actual data, and this is astonishing. I mean, for those of us who've been doing this work for a long time, to have 8,000 independent lines of evidence supporting this intron is really spectacular. The gene that you worked on in the last session, of which Suzanne highlighted in her introduction, was the first annotated alternatively spliced gene in Toxoplasma, done in my lab, as it happens, 30 years ago. And we were able to sequence maybe 10 cDNAs, which was an unheard of number. And here we're looking at more like more like 10,000. There are also predictions for gene models that come from synthesizing this information. And that's true for the Craig gene model predictions that Suzanne had mentioned earlier. If you're interested in further details about the machine learning methods that were used to synthesize this information in making gene model predictions, you can look at a published paper by Axel Bernal from a decade ago or so, which was used for the Craig algorithm. It uses conditional random fields to try to define the most likely versions of this. But I can't overemphasize the importance of this information in being able to assess annotation. And very few sites make this information available. If, for example, you look at the Human Genome Browser or you look at Flybase for Drosophila, what you will see is something like this, what the truth is. And of course, the truth is messy. And if you can't explore the underlying truth, then it may mislead you in the long run. I also do want to point out one other thing under the RNA-seq evidence for introns. We are displaying two tracks here. Those that match the annotation, so we can see that all of the annotated introns are supported by thousands of lines of evidence for this gene. Those for which there is strong evidence, but which have not been annotated, and we've talked about two of the three of those, not another one here. But there are also, in deep RNA-seq data, very many more experimental evidence here that we're not talking about at all. And so there is reproducible evidence, for example, for alternative variants of this last exon supported by 17 different reads, or in this case, by 27 different reads, or in this case, by 517 different reads. But all of these have been excluded from our default view because in the absence of any biological evidence that they should be considered, these are ones that we're suggesting you might want to consider. And the last thing I'll mention about this is that if you care about another species, Cryptococcus, for example, or Aedes, or Giardia, you can run a query on any of our databases to search for genes search gene models for unannotated intron junctions of high confidence. And this is the search that we have run to give rise to to identify the genes that were selected for Toxoplasma here. And you can run this search yourself for species that you are interested in if you would like to be able to explore the accuracy of genome annotation. We are at the end of time for this session, so let me just take a look at, I see there's a comment from Scott Lanfear about the read-through transcripts might have multiple explanations. Some could be cytoscolic transcripts that weren't fully spliced. Some there are a whole so there could be nuclear export problems. You know, absolutely true. As with any of these experiments, this is just this providing this data raises hypotheses that you might want to explore. So, for example, one of the things that oops, sorry, get back here. What we have looked at here gave us evidence that this gene seems to have a fairly high frequency of intron read-throughs, in contrast, for example, to the genes upstream and downstream, which are highly conserved. Presumably what that means is that the selective constraints against that are pretty low. I would predict, for example, that if we look at the phenotypic information for this gene, that this gene is probably not and not highly essential, is not an essential gene, and indeed during the tachyzoite replicative stages, this gene seems to be dispensable. My guess is that the upstream and downstream genes that show lower frequency of that are probably more likely to be essential. But this raises biological questions. Yeah, so this gene is has a mildly deleterious effect when it is deleted in a CRISPR knockout experiment. But good question. So let me ask quickly if there are other questions. We will not have time to explore all of the 27 genes that were assigned to you. But what I would urge you to do is to go back on your own time and take a look at the genes that were assigned to your group and see what you think is interesting. Any particular questions you have, you can type into the contact us link and we're happy to follow up on. But I would add maybe one other suggestion. Keep in mind that all of those genes were flagged up because there was evidence that there are problems or considerations to be made in discussing the annotation. I'd urge you to also always take a look at the gene upstream and the gene downstream, because it's very easy to despair and think that the annotation is just terrible. But in fact, the annotation of most of the genes in at least the Toxoplasma genome and in the Fusarium genome and the Anopheles genome is actually excellent. So I will monitor any questions in the, oh, sorry, one thing, let me respond to this question of Stephen's about unique and non-unique
And David, we do have extra time because of the way the sessions have worked, so we do have extra time. So don't feel
Oh,
like
I'm sorry. I'm
pinched
sorry.
on time.
Actually, yeah, I'm misreading the schedule. We are just at the end of the exercise time. We are now into the discussion time.
Exactly,
So,
exactly.
yeah, exactly. So let me answer Stephen's question. So, so you can imagine, for example, that that short reads might not map uniquely in the genome. And so if we're looking to try to assess the evidence for expression of a gene, and we see and a read can map to multiple locations in the genome, that does not provide evidence that all of those genes are transcribed. It may only be one of that read presumably came from only one gene, and we have no way of identifying which gene that was. So the norm in the assessment of transcript abundance is to map only is to only consider uniquely mapping reads. But one of the things that we encounter in the ViewPathDB project, because we support hundreds of different species with very different biology, is that there are that we need to be able to accommodate that biology. For example, in an earlier exercise today, you were looking at transcription in trypanosomes, where Kinetoplastida famously have very large monocistronic, very large polycistronic transcripts, which are then fragmented into individual messenger RNAs by the addition of a trans-spliced leader. And that means that sequencing the spliced leader RNA is an outstanding method for being able to identify transcriptional initiation sites. Some kinetoplastid species, including Trypanosoma brucei, for example, have a preponderance of tandem gene duplications, even in fairly recent time. So I'll tell you a sort of history that may be of interest to the kinetoplastid researchers here. When we first started mapping RNA-seq data to the genome and wanted to assess the accuracy of that, we looked at a data set for trypanosomes that was looking at both blood stages and insect stages, and we're expecting to see and we're expecting to see high abundance in the insect stages of the, what is it, PARP? Is that the insect transcript? I think that's right. I can't remember. Yeah, the high abundance in the in insect, and we saw nothing. And the reason for that is that this gene was tandemly duplicated and virtually identical, which means that none of the reads mapped uniquely. And therefore, we got an inappropriately inaccurate view that an inaccurate view that there was no transcription in this in this area. So we therefore kept the unique and non-unique mappers as a way of being able to represent what those look like. Another case where this may be common is there was a gene, I think, that we were looking at earlier. I can't remember which gene it was, a gene which showed a small region with no transcript coverage, even though there was also no evidence of an intron spanning read suggesting that that might be an intron. And by turning on the non-unique mappers, we could see that the reason there was no mapping there is that that sequence, it might have just been a stretch of 50 A's, was non-unique and was mapped elsewhere in the genome. So there are reasons for having that evidence there. One of the things that I often do is look through all of the, or encourage people to look through all of the types of data, or even all of the thousands of individual data sets, to ask which of these might be informative for assessing your gene model. For example, the Toxoplasma genome is now very well assembled. So it is now very well assembled. But if I want to look at, let's see, is there a scaffold information here? So I'm now turning on an additional track. Let's get rid of this because we don't need to load it. And I can see that this region of the genome is assembled into one large piece. And if I zoom out, that will probably be true for maybe even the entire chromosome, because the reference Toxoplasma genome is quite well assembled. But for some genomes where the assembly is poorer, I will always want to take a look at the organization of the genome assembly. I'll drag that up to the top and maybe collapse it down because I need to see this, just to alert me that the assembly is good. Obviously, if there's gaps in the sequence that you're trying to look at, the gene models will not be accurate. Any other questions of things that people would like to ask about or highlight? Let me raise one other issue for this particular gene, and I'll zoom back in so that we can see it a little bit further. So something that I have not, another feature that I frequently look at but have not looked at here, or actually maybe I did look at this, is, so it's notable that the intron read-throughs are most common in the 5' end of this gene. And one thing that might suggest is that the translational start site is incorrectly annotated. So for example, we talked about how evidence for skipping this exon or skipping this exon would lead to a frameshift mutation, leading to a misannotation, leading to a dysfunctional protein product. But that would only be true if this is indeed the correct translational initiation site. If there's a long untranslated region at the 5' end and translation starts here, then all of these changes up in the 5' end would really be irrelevant. That was true, for example, for the the gene that Suzanne was showing earlier, the hypoxanthine, xanthine, guanine, phosphoribosyl transferase gene. I'll zoom over to that in just a minute, and while it's loading, we can take a look at it. But in this case, in order to ask whether I am confident of the translational start site, I opened up this gene that we were just looking to, scrolled down to the orthology region here, and looked at individual genes that were involved. And I noticed that there are a series of paralogs for this same gene in Toxoplasma. And I ran a multiple sequence alignment for that here, which shows me that in fact, while there is poor conservation in the N terminus of this gene, there is sequence conservation suggesting that the annotated methionine is probably accurate. But it is also true that those alternatively spliced exons map to this region. And if, for example, translation from this methionine here, which is also conserved, would lead to a functional protein, it's entirely possible that skipping these first 3 exons could still lead to a functional protein product. Let's see. So here we've just moved back to the HGPRTase gene and highlighted information. Oh, actually, I haven't included the splice. Where was that transcriptional start site? I'm not seeing the, don't want to take the time to find the transcriptional start site data, but that indicated that there was evidence for, oh, here, transcriptional start site, it's just not loading for some reason. That gave evidence for transcriptional starts here, but there's clear evidence for transcriptional starts here as well, as you can see from the RNA-seq data. But what looks like an intron read-through here is in fact most likely transcriptional starts that can occur anywhere along this region that will probably be supported by the long-read RNA-seq data. Yes. So this exon is frequently looks like it's read. This intron frequently looks like it's read through. But in fact, what happens is transcription frequently starts in this region. And of course, if transcription starts in this region, there's no way to excise the intron. And none of that has any impact on the mature protein coding sequence because the translational start site is downstream of this. This is a UTR intron. This, incidentally, is something that wasn't recognized and really couldn't have been recognized in the initial publication, which is here from 1 million years ago. We recognized the exon skip polymorphism, and we recognized this as an intron read-through polymorphism. But in fact, that was a mistaken view, a mistake born of assuming that incomplete transcripts had really started at one site, and we just had our sequencing hadn't proceeded to the end. Whereas in fact, there's clear evidence that transcription does start here, leading to this annotation of 4 transcripts that can start here or here, excising the intron accordingly, and include or exclude this exon, which, if you care, encodes a 49 amino acid in-frame exon that is acylated and leads to membrane association of that protein. Okay, more detail than needed. I'm going to read these other comments that come into the chat, and let me see if there are other genes that people would like to take a look at in particular?
So there are a couple of additional questions quickly. I think we can answer them very quickly. One is, are some RNA-seq data displayed as coverage based on read direction and some just combined coverage? And
Yes.
again, that depends on the nature of the data. So if the data is strand specific, and we know that information, I mean, you can pull it out from the alignment, but we align it then as David is displaying as forward and reverse, blue and red. But there will be some data, in particular, older data was not strand specific, wasn't stranded in the actual setup of the experiment. The kits weren't stranded, or they were super expensive to do, and so those are then are combined. And so basically, there'll be black tracks that don't have directionality, or in this case, orange tracks. So depending on the
I've just
database.
this data here for 29 different strains was non-stranded data. And I just went in and turned on 3 of those, and they show up in orange, so they are not strand specific. I have no way of determining whether these 2 genes are on the same strand or on opposite strands.
And the second question was whether you can display the RNA-seq data against other, I'm assuming you meant strains from the same species. And the answer is we typically pick one strain as a reference strain, and we map all the data to. We have some exceptions to this rule where we've mapped the RNA-seq data to a couple of strains when the community requested that, and it was an important thing to do if there are 2 canonical strains that are used by the community. Or sometimes there's a shift between what is being considered the reference genome, and we're not talking about a reference lab isolate, but the reference genome for based on the genome structure and assembly and annotation. And sometimes when that happens, we end up providing the mapping both to the old strain and the new strain. But typically, we only map to a single genome. Now, there are ways where you can do your own mapping and alignments. And later, towards the end of the day, I will demo how you can upload that data. So if you have your own coverage, if you've done the alignment yourself on another strain, you can actually upload your data to the database and explore it privately. And I'll demonstrate some of that later on today. So hopefully that answered your question.
Yeah. So I'm also have pulled up another line of evidence that is actually really useful and important that is rarely available in genome databases, and that is the mapping of proteomics data from multiple different experiments. I have included a track here that shows all MS/MS peptides, and one of the things that it tells me is this is actually this is going to be confusing. One of the things that tells me is for this gene, for example, there is evidence supporting the protein, this protein, and also evidence supporting the annotated intron, but not these 2 alternative introns. The same is true for the HGPRT gene, but in this case, it looks complicated. You'll see peptides that don't actually correspond to the annotated gene. And that is because the HGPRT gene is very commonly used, and HGPRT knockouts are very commonly used as a selectable marker. So there are experiments where this gene has been truncated or deleted, leading to protein recognition in different sites. So this is actually a really misleading example to use. Let's but for other genes that will not apply, and we can see that we can see that case. Let me do one other gene here, and this I am going to do a gene that I did not that we have not looked at, but in our group assignment, we will also ask to take a look at gene 283,540. I have no idea what this gene is. Let's take a look at it with the same configuration we've looked at here and ask whether in something less than half an hour we can get a quick view as to why we were asked to review the annotation of this gene. And once the tracks load, we can see this is a gene that is annotated to have 1 intron. There is strong evidence in support of the protein. The long read RNA-seq data looks to be pretty consistent with that gene. There is a pretty high frequency of read-through for this intron. There is evidence of an exon skip that is not part of the annotation. That exon skip is less than 10% of the total reads. And if we look at the length of those, this goes to location 366 and picks up at location 575. So that would be 209, in this case, -1, 208 nucleotides. It would not be an in-frame protein coding sequence. So I am guessing that, so we can see evidence for that. This really is included in about 15% of the reads, but not across multiple different strains. So I'm guessing that this gene was flagged for consideration as to whether there might be an additional exon that should be annotated in an alternative transcript. And to reiterate, the evidence supports that there really are transcripts that include this exon, which then could not be translated. But but I would argue that this is the equivalent of a biological typographical error. And the abundance of this is so low that in the absence of additional biological evidence, I would not advocate annotating this gene in contrast to some of the other genes that we've looked at. So if there are other comments, please feel free to type them into the chat. But let me maybe suggest that we move over to Uli, who is going to show you